Welcome dear students. Today we are going to discuss an important topic on synthesis, secretion and transport of steroid hormones. The main objectives of today's lecture are to get a brief idea about the steroid hormones, to know how various steroid hormones are synthesized, to describe regulation and release of steroid hormones, and finally we will be describing the secretion and transport of steroid hormones. To begin with, let us first discuss what steroid hormones are. All steroid hormones are derived from cholesterol. Steroid hormones are a group of hormones that belong to the class of chemical compounds known as steroids. They are secreted by three steroid glands: the adrenal cortex, testes and ovaries, and during pregnancy by the placenta. Steroid hormones play a major role in bodily functioning because they are required for many critical physiological processes including water balance, immune functions, survival, metabolism, inflammation, salt stress and development of sexual characteristics. Steroid hormones are commonly measured in patients for the diagnosis, prevention and treatment of hormone related diseases in children, men and women. In mammalian system, there are 6 families of steroid hormones that can be classified on both chemical and biological basis. These are the estrogens, androgens, mineralocorticoids, glucocorticoids, progesterone and vitamin D. These steroid hormones play a critical role in numerous physiological and pathophysiological processes. The vast majority of circulating steroids in mammals come from the endocrine activity of the gonads and adrenal glands, which metabolize the lipid cholesterol to generate the steroid repertoire. Dear students, now that we have understood what steroid hormones are in general, we will now discuss the steroid biosynthesis. Steroidogenesis is a process by which cholesterol is converted to biologically active steroid hormone. 6 classes of steroid hormones, all of which are indispensable for mammalian life, are made from cholesterol via complex biosynthetic pathways that are initiated by specialized tissue specific enzymes found in mitochondria and endoplasmic reticulum. These hormones include glucocorticoids like cortisol and corticosterone, and mineralocorticoids like aldosterone produced in the adrenal cortex, estrogens like estradiol, progestins like progesterone, and androgens like testosterone and dihydrotestosterone produced in the gonads, and calciferols like vitamin D produced in the kidneys. Historically, steroid hormone synthesis only occurs in the steroidogenic glands, that is adrenal glands, gonads and placenta. A significant number of studies have now challenged this view by demonstrating that several organs including the brain, adipose tissue and intestine are also capable of producing steroid hormones. Now, dear students, let's have a look at cholesterol biosynthesis. All these different steroid hormones are synthesized from the common precursor cholesterol and structurally differ only in the pattern of chemical bonds with the rings and modifications on side chains. The cholesterol is the most prevalent steroid in all animals and has multiple physiological roles that include its structural presence in all membranes. Cholesterol also is the starting point in the biosynthesis of all steroid hormones. Vitamin D and the bile acids The level of the total body cholesterol is determined by a complex interplay of dietary available cholesterol, de novo synthesis of cholesterol, and excretion of cholesterol and bile salts. The liver and intestine together account for more than 60% of the body's daily biosynthesis of this important sterol from acetate via a complex pathway primarily found in the endoplasmic reticulum. But most steroidogenic cholesterol is derived from circulating lipoproteins. However, cholesterol is never truly free, as its solubility is only about 20 micromolar per liter, so that the term free cholesterol refers to cholesterol that is bound to proteins or membranes but lacks a covalently linked group. Free cholesterol may be used by the cells or steroid in lipid droplets following reesterification by acyl coenzyme A, cholesterol acyl transferase. Dear students, now moving on to the next, that is glucocorticoid and mineralocorticoid hormone synthesis. The naturally occurring cortisol is the most prevalent member of the family of glucocorticoids and binds tightly to the glucocorticoid receptor, but in vitro it has a high affinity for mineralocorticoid receptor. Whenever the blood concentration of cortisol falls below the normal circulating concentration of 6 to 20 microgram per 100 ml, additional cortisol will be produced. The daily secretory rate of cortisol is 10 to 20 mg per day. In addition, when an individual experiences significant levels of stress, there will be an increased production of cortisol reaching 200 microgram per 100 ml. Cortisol's most important action is to increase and maintain blood glucose levels via the biochemical process of gluconeogenesis in the combined action of muscle cells, fat cells, and liver cells. Mineralocorticoids are a class of steroid hormones that regulate salt and water balances. Aldosterone is the primary mineralocorticoid. Mineralocorticoids promote sodium and potassium transport, usually followed by change in water balance. Cells of the adrenal, zona fasciculata and zona reticularis synthesize and secrete the glucocorticoid cortisol as depicted in the figure. The adrenal zona glomerulosa cells preferentially synthesize and secrete aldosterone, also shown in the figure. Dear students, now moving on to the estrogens and progesterone hormone synthesis. The two most important steroid hormones of the adult female are 17-beta estradiol or estradiol and progesterone. In addition, two metabolites of estradiol, that is estrone E1 and estriol E3, circulate at high levels at certain phases of menstrual cycle and during pregnancy. E1 and E3 have been thought to be the inactive metabolites of estradiol, but E3 has significant effects on the immune system, and a closer examination of the physiological functions of these two steroids is warranted. As for androgens, the biological actions of E2 can be divided into those directed towards the development and maintenance of the female reproductive system, and those that have effects on several other organs, including the cardiovascular system, metabolism, and brain. The naturally occurring estrogens are typically 18-carbon steroids that have an aromatic air ring with a phenolic hydroxyl. The naturally occurring progestin, that is progesterone, has 21 carbons with another one additional oxygenation, that is oxo, on both C3 and C20 positions. Ovarian estrogen synthesis, as reflected in plasma hormone levels, pulsates every month during different phases of the menstrual cycle and changes throughout the life of a normal human female. The peak of estrogen synthetic pathway during the 6 months after birth is followed by the relative quiescence throughout childhood until the beginning of the pubertal period. Plasma estrogen pulsates at adult levels by the end of puberty and begins to decline in middle age, that is menopause. The amounts of estradiol, estrone, and progesterone produced by the ovary and circulating during different phases of menstrual cycle. The enzymatic steps of estradiol synthesis are partitioned between the granulosa and theca cells of the ovary, which surround the oocyte and form a follicle as shown in the figure. All circulating estrone and estradiol are produced by the aromatization of androgens, including those derived from adrenal and placental steroidogenesis. In addition, P450 aromatase is expressed in non-steroidogenic tissues, especially fat and bone. As a whole, in order for sufficient amounts of estrogen to be synthesized for maturation and ovulation of the follicles, both granulosa cells and theca cells must be functional. Following ovulation, the major steroid produced by the luteinized cells of the corpus luteum is progesterone, although estrogen continues to be synthesized and secreted as depicted in the figure. Dear students, now coming to the synthesis of vitamin D. Vitamin D and its metabolites are not technically steroids in the strict chemical sense, as the B ring of cholesterol is opened, corticosteroid. Nevertheless, these sterols are derived from cholesterol, assume shapes that are very similar to steroids, and bind to a nuclear receptor. In the human skin, ultraviolet radiation at 270 to 290 nanometers directly cleaves the 9 to 10 carbon-carbon bonds of the cholesterol B ring, converting 7-dehydrocholesterol to cholecalciferol, that is vitamin D3. Plants and yeast produce ergocalciferol, vitamin D2, which has essentially the same properties as cholecalciferol. Both calciferols are biologically inactive prohormones that are then activated and subsequently inactivated by mitochondrial P450 enzymes. We will now discuss about the control of steroid hormones and their release from the respective glands. The production and secretion of most hormones are regulated by the homeostatic mechanisms which operate in that particular endocrine system. The secretion or release of the hormone is normally related to the requirement for the biological response generated by the specific hormone. Because of their hydrophobic nature, steroid hormones and precursors can leave the steroidogenic cell easily and are not stored. Thus, steroidogenesis is regulated primarily at the first step in their synthesis, that is the cleavage of the side chain of cholesterol, and at the level of steroidogenic enzyme gene expression and activity. Once the biological response has been generated, the secretion of the hormone is restrained to prevent an over-response. Thus, a characteristic feature of most endocrine systems is the existence of a feedback loop, that is either direct or multi-step, that limits or regulates the secretion of the hormone. Another contributor to the biological availability of steroid hormone is the control exerted by biological rhythms on steroid hormone production. Steroid hormones are synthesized but not stored within specific endocrine cells from which they are easily secreted in the plasma. However, most steroid hormones have limited solubility in plasma due to their intrinsic hydrophobic nature. Accordingly, steroid hormones are largely bound to specific plasma transport proteins which are synthesized in the liver. All steroid hormones except aldosterone have their cognate plasma binding proteins. The excess of steroids that are not released or bound to the receptors will be inactivated into the cells and hormone metabolites will be secreted in the circulation. This local inactivation of the excess of active steroids inside the cell is an important manner to regulate active steroid concentration. As a whole, the key determinants of the steroid hormone responses are the presence of a response system, that is receptors and signal transduction pathways in target cells, and hormone blood concentration. The effective concentration of a steroid hormone is determined by the rates at which the steroid is biosynthesized and enters the body pools, the tightness of binding of the steroid to its plasma carrier protein, and the rate at which the steroid is biologically inactivated by catabolism and removed from the body pools. Dear students, we will now discuss steroid hormone secretion and transport. Steroid hormones, like other hormones, are chemical messengers that send a signal within a physiological system from point A, that is point of secretion, to point B, that is point of biological action. Steroid hormones are synthesized but not stored within specific endocrine cells that could be associated with an anatomically defined endocrine gland. Upon the receipt of an appropriate physiological signal, which may take the form of either a change in the concentration of some component in the blood, example that of another hormone, calcium ions, stressors, or a neural signal, the hormones are released into the circulation. They are then transported into the bloodstream to one or more target cells, which are defined as targets by the presence of the specific high affinity receptors, members of nuclear receptor superfamily located either on the membrane or within the cells, that is endocrine system, as is shown in the figure. A type of hormonal communication system does not involve the circulatory system at all. In paracrine systems, hormones secreted from the steroidogenic cells interact with their cognate receptors in neighboring cells, which are reached by diffusion as depicted in the figure. As with endocrine system, the nearby target cells may be all the same type or may differ from each other. Several, if not all, of the steroid hormones act by paracrine in addition to endocrine mechanisms. For example, in the testes, testosterone not only is released into the blood from the interstitial cells in which it is produced, but also diffuses to nearby seminiferous tubules to support the production of sperms. Finally, some cells both produce the same hormone and respond to it. This type of system is referred to as autocrine for all class of hormones, excluding steroid hormones. The appearance during evolution of the repertoire of cells specific steroidogenic enzymes permits to produce steroid hormones intracellularly according to the local needs without biologically significant release of active sex steroids in the circulation. This type of system specific for steroid hormones is referred to as intracrine as depicted in the figure. Examples of endocrine system involves androgen or estrogen synthesis in non-steroidogenic tissues during andropause and menopause. All tissues except the endometrium possess the intracrine enzymes able to transform dihydroepiandrosterone or DHEA into androgens and estrogens. Humans, along with other primates, are unique among animal species in having adrenals that secrete large amounts of the inactive precursor steroid DHEA, which is converted at various levels into active androgens and estrogens in specific peripheral tissues according to the mechanisms of intracrinology. It is very important to mention that an essential aspect of endocrinology is that the active sex steroids are not only made locally, but that they are also inactivated locally at exactly the same site where synthesis takes place. In fact, the sex steroids made from DHEA in peripheral tissues are essentially released outside the cells as inactive compounds. DHEA of adrenal, ovarian, or exogenous origin is distributed by the general circulation to all tissues indiscriminately. The transformation of DHEA into estrogens, androgens, however, is tissue specific, ranging from none in the endometrium to various cell specific levels in the other tissues of the human body. Most importantly, approximately 95% of the active estrogens and androgens are inactivated locally before being released into the blood as inactive metabolites, thus avoiding inappropriate exposure of the other tissues. As a whole, the intracrine process that is typical for steroid hormone binding to nuclear receptors is thus equivalent to autocrine and paracrine processes activated by hormones binding to transmembrane receptors. Although the terms autocrine and paracrine are used for both nuclear and transmembrane receptors, it is important to make the distinction, especially because nuclear steroid receptors could be also found extrinsically linked to the plasma membrane and act in an extranuclear manner. Dear students, it was all about today's lecture. Hope you have understood it well. Thank you. Goodbye.